So if I were to put the names of these things up on the board, I'm sure you would be able to match. That's easy. What's hard is to come up with the name. What's harder is to come up with the name of a smell sometimes. So what I would like you to do is somebody tell me, what is one? Just one word. Olivia. Pine. 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 Anybody come up with anything else? Did anyone say things like, oh, it smells really familiar. I know that smell, but I can't come up with the name of it. Right? That's pretty common to say, oh, yeah, that smells like a forest, or that smells like a tree, but not to come up with the fact that it is in fact pine. Okay, so identification of odors is interesting. Two, what did people have for two? So even a little bit more challenging than pine. Anybody? Yes, Jenna.
Rose.
Rose. Rose smell. What commonly happens in this room when they smell rose is I can't place it, but it reminds me of my grandmother or somebody else who wears floral perfume. Your grandparents may not wear floral, maybe your great-grandparents. Floral perfumes are falling out of favor. But I mean, sometimes you've been in an elevator with somebody who's just wearing overpowering gardenia scent, and it's just like, oh my God. So that's rose. That one was challenging. So Jenna got it. I bet others of you are saying, oh yeah, right, but didn't have that at your fingertips. Three. What is three? Anybody? Get three? Give me, so three might be maybe more challenging than rose. Anybody got a name? Just a word. There's no right or wrong answer here. This is an experiment of what word you can come up with for this smell. Olivia? Cinnamon. Yeah. Okay, cinnamon. Anybody else? That's not right, but it's close. This is clove oil. So it's the smell of spice. Cinnamon and clove are commonly used together in cookies and things like that. So this is the smell. Clove is similar to, but distinct from. If you put cinnamon and clove next to each other, and I told you one is cinnamon and one is clove, you wouldn't have any trouble telling them apart. But you have trouble coming up with the name of a smell. And the last one, four. Four is, I think, easier. Well, maybe not, because it's not a strong smell. Anybody got a name for four? Citrus. It's actually lemon. So you thought it was maybe mixed citrus, orange, lemon, grapefruit. It is lemon scent. So you were able to identify citrus, because that's close, but not exact. So you can see we have a pretty good sense of smell. That is to say, we can detect things at very low concentrations. But identifying things without other cues is pretty hard. So what we're going to do now is talk about the chemical senses. We detect chemicals with our nose and with our tongue. Part of olfaction and gustation, or taste. Smell and taste. And we're going to talk about the molecular mechanisms of those senses. Okay, let's focus on smell first, the olfactory system. Obviously, smell happens, our sense of smell happens in the nose. We don't have any chemical, we don't have very much chemical sensors in our fingertips. Nasal cavity. At the roof of the nasal cavity, the superior edge of the nasal cavity, is the olfactory epithelium. And this is where we're going to transduce a chemical into an electrical signal that our brain can interpret. So the olfactory epithelium is going to be connected to the olfactory bulb just on the other side of the skull. So there's going to be cells that are facing the inside of the nose, the nasal epithelium, and they're going, well, we'll see. Then there's the olfactory bulb just on the other side of this bone. That's going to, and then the olfactory bulb is connected to the olfactory nerve, the cranial nerve 1, which goes to the brain. So here's a blow-up of the superior part of the nasal cavity, the olfactory epithelium, bone, and then to the olfactory bulb. I think the next one has another one. Olfactory epithelium, lining the inside of the nasal cavity, bone, perforated bone, because nerves go through it, and make synapses on glomeruli in the olfactory bulb, which is sitting on top of this bone. These cells, these are the cells that do chemical transduction in the olfactory system. They have cilia that are embedded in the mucus of the nasal cavity. So there's mucus here all the time, even if you're not, don't have a runny nose, even ordinarily there's mucus. Cilia are sitting in that mucus. The cell bodies are embedded in this epithelium that is connected to bone, and axons from these neurons embedded in this epithelium, axons make synapses in the olfactory bulb. So now, is the olfactory system, are these long or short receptor cells? You got a 50-50 chance of getting it right, just tell me. Well, you're better than 50-50 because you might know. Detector, receptor, axon, synapse. Long or short? Long. It's not a very long cell. It's going from one side of the bone to the other side of the bone. It's not as long as a somatosensory detector, but it is a long. We classify these as long receptors because they have axons and they fire action potentials. So what do I want you to know from this? Cilia is where the odorants are going to bind. Olfactory receptor cells, axons making synapses in the olfactory bulb. You do not need to know the name, the word glomerulus. You do not need to know the name mitral cell. But you do need to know olfactory bulb, mucus, olfactory cells. I do not force you to learn the name of this bone. It's got a name, but I'm not asking you to learn that. So we detect chemicals. That's what we're doing when we detect smells. We're detecting chemicals. And we call them odorants if they smell. And so, here's just a few of the thousands of different odorants we can detect. This molecule smells like almonds. This molecule smells like skunk. There's the smell of rose. There is the smell of ginger. That's what clove, what clove smells like. That's what it looks like chemically. Here's an interesting one. We can detect different stereoisomers. So that our stereoisomer of this molecule smells like mint, and I've got spearmint up here if you want to refresh your memory of what mint smells like, but you chew gum and it smells like that. The S stereoisomer of the same molecule smells like rye bread, caraway seed. So we can discriminate pretty clearly. Spearmint does not smell like caraway. we can discriminate stereoisomers. Some of them, anyway. So there's no commonality. Well, there's a few commonalities. Skunk, all the skunky smells have this thiol. If you're smelling a skunky smell, it's got a thiol. But the rest of them, you can't make a correlation between, here's an aromatic ring that smells like clove, and here's an aromatic ring that smells like almond, and here's an aromatic ring that smells like ginger. You could say, well, okay, maybe this is spicy, this is spicy. Is this spicy? I don't think you smell almond as spicy. Although we make cookies with almond, ginger, and clove, so I don't know, maybe. Anyway, there's no clear relationship between the chemical structure and the smell. How do we detect chemicals? Well, the working end is the cilia. That's what we're zooming in on now. We're zooming in on the cilia that are being contacted by odorant molecules. So this is the outside of the cilia. This is mucus out there, and this is the intracellular compartment of the cilia. So we're in the cilium. Mucus, intracellular. There's the odorant. What type of receptor is this? Is it a ligand-gated ion channel? No, it's a GPCR. All of the odorant detectors are GPCRs. And they go through the GPCR story that you now know very well. Binding, conformational change of the alpha. Alpha lets go of its GDP and binds GTP. In a cartoon like this, just to be clear, one more time, this is not GTP going to GDP. This is GDP coming off and GTP binding. GTP binding, conformational change, separation of the alpha from the receptor, separation of alpha from the beta gamma. Same old story. The name of the G protein here is G-olf. It's not golf, but it's G olfactory. It's got a name. So it's not GS, it's not GQ, it's not GI, it's not transducin, it's G-olf. Remember, transducin is the G protein in the visual system. In the olfactory system, it's called G-olf. But everything about it is the same as the G protein story you've learned. This activated G protein activates adenylyl cyclase. You've seen this before. So G-olf, of all the G proteins that you've learned before, G-olf is most similar to which one? Transducin, GI, GQ, or GS? GS, because it activates adenylyl cyclase. Okay, so now we're sort of in the GS story. Adenylyl cyclase converts ATP to cyclic AMP. You have learned that cyclic AMP interacts with the regulatory subunits of protein kinase A. That's true. But in the olfactory cilia, cyclic AMP activates a cyclic AMP-gated cation channel. You've seen this story before. Activation of a cation channel, sodium and calcium influx, depolarization, threshold. Remember, we're in a long cell. It's got an axon. It has sodium channels. We don't go straight to calcium channels in this case. We go to sodium channels, action potential frequency. Action potentials carried in this axon by the mechanisms you've learned about, even though this is only a quarter of an inch, it's still a distance. Action potential propagation, And now we have the voltage-gated calcium channel story and neurotransmitter release in different sections of the olfactory bulb. Okay? So this is how we detect chemicals, odors. It's a pretty simple, straightforward story using some of the building blocks that you already understand.
Bob?
Yeah.
So inside the cilial cells, it doesn't look like there's very much deviation in that kind of G protein pathway. I mean, there's like 10,000 different kinds of receptors.
We're going to get to that we're going to get to that right now.
Oh, okay.
So just hold that question. We do not have 10,000 different types of receptors in order to discriminate 10,000 different odorants. You'll see in a moment how we do that. Zoe.
How are taste and smell related?
So taste, you're going to see in a moment, taste has, so olfaction has about 10,000 different modalities. 10,000 different odors that we can discriminate. Especially if you're given clues, like, which one is fruitier, or which one is more rancid, or things like that. We have 5 tastes, only 5. And so taste is very simple. Salty, bitter, sour, delicious, umami, and sweet, of course, sweet. That's what we detect with our tongue. All the other richness of flavor comes from odorants in our food that go from our tongue, in our mouth, into the nasal cavity and are detected as smell. So people who have lost their sense of smell, either because of COVID or for other reasons, they have really depleted sense of taste as well. It's unpleasant because we lose our sense of smell and the world becomes a little bit paler, just less robust of an experience. But you lose your sense of taste, and that's a problem. I mean, it's really hard to enjoy food if you can't taste it. And if all you can taste is salt and sweet, bitter. Anyway, flavor. So what you're getting at, Zoe, is that flavor is a mixture of taste and odor, and olfaction. Okay, so we've done olfaction. Well, no, we're not done. Different receptors connect to different bulbs in the different glomeruli in the olfactory bulb. Now, let me answer, I think that was Vincent's question about how do we detect 10,000 different odorants. We don't have 10,000 different receptors. We have on the order of 350 different odorant receptor molecules that have different binding properties. So they bind, they bind one odorant very well, and they bind another odorant very poorly, and in between. So 350 different odorant receptor molecules are expressed in our olfactory cilia. We're a little bit small compared to a dog and a rat, but not too bad. Those 350 different types of receptor molecules are expressed in millions of receptor cells. So we're pretty small on the receptor cells. We have only 12 million receptor cells, and a bloodhound has 4 billion receptor cells. So this is an animal that has evolved and then through selection evolved to have an extremely sensitive sense of smell. So we have 350 different receptor molecules. They are all GPCRs. There are variations. So this is just showing you the 7 transmembrane helical structure of a GPCR, and some of these amino acids are highly variable, and some of these amino acids are highly conserved. And the variable ones in these different 350 different versions of this is what gives them their different selectivity for odorants. But now, how do 350 different receptors detect something like 10,000 different odorants? And the answer is that we use combinations of different receptor activation to give us this discrimination. So for example, let's just find a simple example. Well, let me read the quote from Linda Buck, who won the Nobel Prize, who shared the Nobel Prize with a few other people figuring this out. Each receptor recognizes multiple odorants. So each receptor detects multiple odorants, but each odorant is detected by a unique combination of receptors. So here is a receptor that's activated by C6. Well, let's go to the C8 group, because that's what I've translated here. C8. So we have C8 carboxylic acid, which is octanoic acid, which smells fruity but sour. So C8. C8OH is octanol, and that smells citrusy, fresh. And octanal, I have octanal on here. I guess I don't. Octanal smells just generally citrusy. The point is, here is a receptor that's activated by carboxylic acids of C6, 7, and 8 length, and this other one. So it's activated by this set of odorants. Here's another one that's activated by this set of odorants. And here's another one that's activated by this set of odorants. It's this combination that would allow us to say it's this set of smells. So the combination, so let's just see. This smell, whatever this smell is, bromo C7 carboxylic acid, whatever this smell is, it smells distinct from the others because it is activating this population of receptors in, some of them strongly and some of them weakly. This combination of activation is what allows us to detect this odorant specifically. We don't have one that's detecting this odorant. We have multiple of them that's detecting this odorant, and it's the combination that allows us to discriminate 10,000 different odorants. This is the relevant quote. Each receptor recognizes multiple odorants, but the combination is what makes things unique. Did that answer your question, Vincent? I think it was.
Yes, absolutely. Thank you, Bob.
Okay. So information is transduced by the combination of activation of these receptors, by the combination of synapses in the olfactory bulb, and then by information carried from the olfactory bulb to the brain. One thing that is unique about the olfactory system, so this is unique, so I could definitely ask you this. One thing that is unique is that the olfactory information is not processed in the thalamus. It goes straight to the brain. All the others have a link in the thalamus, processing and relaying in the thalamus. Olfactory goes straight to the brain, and it goes straight to the olfactory cortex, which is it's in the medial temporal lobe. So here's the superior temporal gyrus. This thing is pulling away at the lateral sulcus to reveal the medial temporal lobe. What else? What's another structure that's in the medial temporal lobe that I've taught you about?
The hippocampus.
Yay, the hippocampus. So the olfactory cortex is very close to the hippocampus, which at least partially explains why smell and memory can be so tightly linked. Smells can evoke a memory stronger, then, I mean, sound can, sir, and, obviously seeing something can trigger a memory. But we're pretty good at remembering something, not necessarily remembering the smell, but remembering something related to that smell. So for some of you, the smell of cloves and cinnamon might just remember, cooking, baking cookies with your mom, or something. That trigger is because the olfactory cortex and the hippocampus are coupled pretty tightly, both physically and electrically. So I'm going to skip the next slide. I mean, once we activate the olfactory cortex, the information goes everywhere in the cortex. That's what this is. It's not important to know where it's going because it's going everywhere. We're processing that information in our brain. Questions about olfaction. Okay, now you know something about how we turn a chemical into the sense of smell. You don't have to. You don't. Now let's turn our attention, let's take a 5-minute break right here. We'll change the video, and we're going to turn our attention to how we detect taste. Now remember, flavor is a combination of taste and smell, but we need to understand taste as well. So we're going to get to the other way that we detect chemicals by taste. Let's take a 5-minute break, and then we'll get to that.